The cell cycle. Interphase. In interphase, the nucleus of the cell is well defined and surrounded by the nuclear envelope. The nucleolus, the dense region in the nucleus that creates the building blocks of ribosomes, is well defined and visible in interphase. Centrosomes, an organelle that organizes microtubules, are replicated in interphase during the S phase. The original one centrosome replicates into two. During interphase, the cell grows and prepares to divide. Prophase. In prophase, the nuclear envelope dissolves, meaning that the nucleus of the cell is no longer defined and the nucleolus disappears. The chromatin, a complex of DNA and protein, found originally in the nucleus, condenses into visible chromosomes. A chromosome is comprised of two identical sister chromatids, which are connected at the centromere. At the centromere, the kinetochore is also found, which is the area of the sister chromatids where microtubules attach. Metaphase. In metaphase, microtubules grow from the two centrosomes, pushing them to opposite ends of the cell, a process that is initiated in prophase. The condensed chromosomes line up at the metaphase plate, which is the equator of the cell. The microtubules that grow from the two centrosomes then attach to each sister chromatid at the kinetochore. This entire microtubule apparatus is also called the spindle. At the end of this phase, chromosomes are at the equator of the cell and the microtubules are attached to each sister chromatid. Anaphase. In anaphase, the centromeres separate, allowing for the microtubules to retract as they are shortened, taking the separated sister chromatids, now called chromosomes, with them. This causes the chromosomes to move to opposite sides of the cell. Telophase. In telophase, two new nuclei form, one at each end of the cell. The chromosomes uncoil and spread out, turning back to loose fibers called chromatin. Mitosis, which is the division of one nucleus into two identical nuclei, is now complete. Cytokinesis. Cytokinesis is when the rest of the cell, such as the cytoplasm, cell membrane, and organelles divide. This process happens simultaneously with telophase. In animal cells, proteins called actin and myosin create a contractile ring around the middle of the cell. These proteins pull at the center of the cell, acting like a drawstring, creating the cleavage furrow. This pinches the cell in the middle, and the drawstring, which is a contractile ring formed by actin and myosin, pulls so tight that the cell splits into two. The original cell has now divided into two identical cells, and replication is complete.